Dear Humboldtians, dear guests, my colleague Carmen Klein and I would like to welcome you to the second day of the annual meeting and to this keynote lecture. I'm very glad you found way into this room to the keynote, Bridging the Diversity of Ideas, about biodiversity. My name is Monica Lieb, and I'm head of the Division Europe in the Sponsorship Networking Department of the Alexander von Humboldt Foundation. And my colleague Carmen Klein is Program Officer in the Division North America, Australia, New Zealand, and Oceania. And please let me make a few technical remarks before we actually start. In case you have technical problems, the usual advice, please try to refresh your browser or leave the room or even the event and try to return. And if this doesn't help, please turn to the help desk, which you will find in the upper hand right corner with the 3 dots under More. You will find the help desk. One important note is this keynote will be recorded. So the slides of Professor Leibold and everybody who has their cameras on will be recorded, will be seen on the slides. The chat will not be visible in the recording. All your cameras and micros are switched off at the moment during the talk, but later on we can give you broadcast permission, so you can put on your camera and your micro to ask questions or comment on the lecture. And you can also use the chat for comments even during the lecture, and we will get back to them later. So we will try to stay in contact with the chat and see what's going on. But now let's get started. I have to find out now my mouse is gone. I made such a nice note. Okay, yeah, it is my pleasure. I knew that to introduce Professor Mathew Leibold from the University of Florida in Gainesville. And I had to look it up a little bit because Professor Leibold's CV is almost as diverse as his research topic. Being half French and half American, he told me yesterday, he grew up in Spain. And he has been all around the United States during his scientific life. He was in Arizona, in Michigan, North Carolina, Illinois, and since 2018, he is a full professor in Florida. In 2017, he received the Humboldt Research Award, and his nominating colleague Jonathan Chase in Halle, he wrote in his laudatory address, "Professor Leibold is an international authority in community ecology. He has published foundational studies from niche to ecosystem, but his influence goes much farther. He makes people around him better scientists." And I do believe this after I've spoken to him. Today, he will share his ideas with us, bridging the diversity of ideas about biodiversity. Please, Mathew, now the floor is yours.
Yeah, thank you very much. And thank you for inviting me to give this keynote. It's really an honor and a privilege to do so. Alexander von Humboldt is, I consider him the grandfather of ecology. I'll talk about the father later. But his observations in his trips and his observations about nature, and especially about the distribution of vegetation, are foundational in ecology, especially the idea that the elevational patterns in vegetation correspond to latitudinal patterns, but many other aspects of his work. I'm very happy to be part of the Humboldt family. And I found out a colleague after I was nominated for the award, or after I was awarded the award, told me that I was a 7th generation academic grandchild of Alexander von Humboldt, which turned out to be true. And so I'm in the Humboldt family in 2 ways, and that's really great. Anyway, just to connect Alexander von Humboldt with biodiversity, of course, he collected specimens and organisms in various parts of the world, especially South America. But what makes him stand out more is that he linked these to observations and made ecological inferences that are, quite modern. One of the ones I really like is he observed that monkeys in the Amazon would just, when they heard a leopard, would just stay home and not go out to feed, even though they might be hungry. And this seems maybe somewhat trivial, but in recent years, that idea has been expanded into a whole line of research called landscape of fear, in which this is not just limited to monkeys, not just Amazonia, but a general phenomenon that affects organisms of all types, including protozoans and such. The other thing that I think is remarkable about Alexander von Humboldt is, especially later in his life, he tried to organize and interconnect his ideas in an integrative way, especially in his book Cosmos. And I think this is one of the most marvelous things that he did. So let's talk about biodiversity a little bit. It seems like a simple thing to think about. You go out into nature and you start making lists of species, or you collect them like Alexander von Humboldt did, and you end up with a lot of them. And so you go, wow, there's a lot of these out here. And you could call that biodiversity. But it's quite a bit more subtle. So, for example, each species can have genetic diversity within it. And that diversity might be mixed everywhere. There's always a lot of genetic diversity, or it might be structured. The genetics in one place are different than another. Of course, oftentimes that's linked to trait diversity. So a bird may have a big bill in one place and a small bill in another, or even in the same place, some birds have big bills or small bills. And this is exemplified by the work on the Galapagos finches that was done. Beautiful studies. Oops, what happened there? So, I'm not sure. I didn't do that. That means that these organisms may also differ in functions, or we might be interested in how they're related to each other. The other important thing is that biodiversity can vary with scale. So the number of species that you find in one site, of course, if you go to multiple sites, you will see more of them. And so you have to define biodiversity by the scale you're thinking about. And the same thing is true with the time scale. So I study mostly boring plankton. It's not that boring. But if I go to a lake and I dip my net in there, I will get a almost instantaneous assessment of biodiversity. But if I keep going week after week, I will find new species. And I may do that over an annual cycle, which makes sense because many of these species have annual cycles. Or I could persevere and do it over decades, and I will find other patterns. The other intriguing thing to think about biodiversity is interaction biodiversity. What I mean by that is that these species interact with each other. And so in one place, a bird may, for example, only interact with one plant species, and that's the only thing it eats. But that same bird species may, in another place, given access to other plants, interact with multiple plant species. And so the number of interactions could change. So it's quite a subtle and complex idea, much more so than just counting the number of species. Why do we want to think about biodiversity? Well, one of them is that this complexity itself is really fascinating. And, I have to admit that for me, this is my primary reason. How do we understand these various patterns that I've described to you verbally, and how do we quantify them, and how do we link them to hypotheses about mechanisms? The other one is that biodiversity may have important implications, and some of these implications could be really important to humans. For me, the most obvious one is the aesthetic one. I just think nature is beautiful, and biodiversity provides ecosystem services to me of beauty. But, not all humans are quite wired the same way I am. Many of them would prefer to have diversity of movies or books or something else, and they may not care about all this Humboldtian biodiversity aesthetic. But even for those people, biodiversity could be important because it provides ecosystem services. So, for example, biodiversity may influence carbon sequestration or pollutant clearing in water or soils. And it may buffer against perturbations such as climate change. And what that makes us realize is that biodiversity is actually part of a bigger cycle that we can think of as the coupled human-nature system. So the coupled human-nature system is a relatively new idea. When I was applying to graduate school in the early 80s, I knew that I would be interviewing prospective advisors, and they would ask me, why do you want to be an ecologist? And the wrong answer was to say, because I want to save the planet. That answer would result in my prospective advisor saying, oh, that's wonderful. Yeah, let me make some phone calls and introduce you to some people in the wildlife department who will love to talk with you. And I would not have been working with them. Instead, at that time, community ecologists studied natural systems, and they struggled very hard to find them and ignore the humans in the picture. And so they would go to remote places on Earth or try to find closer by places that, weren't affected by humans. That changed in around 2000, I think, in a dramatic way, so that now virtually all ecologists have some component of their thinking that relates to this coupling of humans and nature. And the important thing about this coupling is that it's reciprocal. So it's not just that humans affect nature, but that there's feedback that affects humans. And that this coupling in itself is also an important thing to study. I don't find it as beautiful, but some people do, and that's good. So in biodiversity ecology, one could argue that we're at a critical transition. How do we develop our growing understanding of biodiversity in ways that help us understand this coupling? The problem is that we've learned a lot, but our knowledge is not up to the task. It doesn't address biodiversity sufficiently well at the huge scales that biodiversity actually operates in nature. And we need to solve this problem. We need to be able to address these questions because these are the ones that are going to affect human welfare and the conservation of even whatever natural systems we have remaining. So the present situation, and the reason I think it's a critical transition, is because we are starting to have tools that can help us, and that's what I'll try to talk about. And the question is, can we bring them into action sufficiently early to make an impact? So just to kind of put a historical bent on this, I'm kind of a fan of history, so I like to know where we've been before I figure out where we're going. We've already talked about Alexander von Humboldt and the other naturalists who wondered and collected and classified this biodiversity that we're interested in. But their ideas were not very well developed. Probably the first great ideas that came to affect biodiversity were the ones of Darwin and Wallace in the development of evolutionary theory. And it's important to realize that what they did was to look at slow time and large scale aspects of biodiversity. And that was great, but it didn't quite address all of the complexities that we're interested in. The person who I think was the most critical in developing ecology, and he is the person I think of as the father of ecology, is Charles Elton. And there's a picture of him here on his motorcycle at the time that he wrote his book. So he was 27 years old, and he wrote this book called Animal Ecology. And in it, he introduced 4 of the major conceptual ideas of modern community ecology: the concept of niche, the concept of food web, the concept that body size is a hugely important trait, and the concept of trophic pyramids. And the critical thing about his ideas was that they happen on fast and local scales. I'm ignoring a lot of very important ecologists, but I want to emphasize Robert MacArthur, because he was the person who most fully made the connection between these ideas that were verbal ideas and mathematical theories that could capture the essence of these ideas, and thus developed the theory of ecology. So mapping ideas into equations and to expand our ability to gain insights from these ideas was a tremendously important thing. The school of MacArthur flourished with lots of ecological theory. Unfortunately, that theory kind of got ahead of the data, and a reality check was provided by Simberloff and Strong in the late 1970s. And they introduced the idea that if we're going to test these ideas that are really cool, we should have null hypotheses against which we're going to try to be rigorously statistically inferential. And their early generation models, let's call them Gen 1 models, mostly rejected most of the predictions of the MacArthur School. And this is about the time I became a graduate student. Community ecology was in, or biodiversity ecology was in a crisis because there was evidence that very few of the ideas, theoretical ideas, were substantiated. We have theory, we have observations, and we want to know, does this make sense? The alternatives were nature just doesn't care about our theories at all, or we just don't understand how nature actually responds to our theories. And so by conducting experiments, we could try to assess that. I have put here pictures of 5 of my favorite experimental ecologists. I won't explain my choices, except to point out that the guy in the blue shirt was my PhD advisor, and was hugely influential to me, both personally and professionally. His name's Earl Werner. The other development that happened was the development of more sophisticated statistical models, and that also meant that we could try to be more subtle in the patterns that we might hope to detect. And here are 3 of the people that I've been influenced by. So right now, so we can think about a little bit more recent work that has happened since since this time. There's 2 things that are important. One of them is the idea of community assembly. And this is the idea that, yes, we have a regional pool of species, but when we look, so we have Humboldtian regional pools of species, but if we become Eltonian and we think we want to look at a single spot, we have to think about how we get what the particular subset of species that we find. One approach is to think about these filters. And one of the people that developed this approach, and he's not the only one, is Paul Ketty. And so, in this conceptualization, we have a regional pool. One thing that happens is that not all the species in this regional pool may have equal access to all the local sites. And so there's a dispersal process, limited dispersal, that can act as a filter. A second filter is the environment. So, for example, we may have cactus and cattails in the same regional pool, but we don't expect, even if they both have access to a desert and a water, a pool of a pond, we don't expect the cacti to grow in the water of the pond, or the cattails to grow in the desert. And that's got nothing to do with anything else. The third filter is what they called the biotic interaction filter. And here, the idea is that the species that happen to be together due to their interactions may also act as a filter. So some species may outcompete other species or depend on them, etc. The alternate framework for thinking about community assembly was one that was developed by, at least I associate it mostly with Richard Law. And this looked at the process of community assembly as an iterative process of colonization. A single species arrives, it interacts with whoever's there, things shake out, and then maybe some things go extinct or maybe not, and then there's a transition to another, the possible transition to another. So the simplest case is shown here with this orange square, which is empty, and so that's an empty patch. It could be colonized either by an orange square species, which means that it's a good fit to the environment, or a blue circle, which means, yeah, it can live there, but it's not really all that good at it. And so those are two possible transitions. Of course, each one of those could go extinct again and return it to the empty state. And then the third thing that can happen in this case is that the blue species may get there first, but then when the yellow species arrives, it kicks it out. And so you can see that in this little assembly graph, this graph is called an assembly graph, you end up with inevitably more likely to be occupied by the square yellow orange species than by the blue species, but you may find the blue species in some patches before it is outcompeted by the orange species. That's kind of so that's an important and interesting way to think about community assembly, because here it's kind of trivial, but if you start having lots and lots of species present, it can become much more interesting. So for example, one thing that Richard discovered was that you may not end up with a single best fit species. You may end up with what's on this right-hand panel. These are called assembly cycles. And here, what happens is each time there's a transition, it changes, and some species go extinct and then get replaced. And then eventually it comes back to that same species pool. And so if you were in a pond or a place that followed these rules, you would continuously see this cycle, kind of like a rock, scissor, paper kind of game that would cycle through time. The other important concept, and the one that I was so lucky to be involved with, was the meta-community idea. Here, this was an attempt to link the feedback between local and regional effects by thinking of ecology systems as meta-communities, meaning lots of places that are connected by dispersal. I'm going to kind of breeze through this a little bit faster than I would like, but we worked on this mostly as a working group sponsored by NCES, which was maybe the most important influential synthesis center in ecology, and has led to other synthesis centers such as IDIV in Leipzig. So here's some of my collaborators in that group. You'll recognize Richard Law there, and John Chase, who's at IDIV, Priyanka Amarasekara from Sri Lanka, Marcel Holyoke from England, and so is Richard, and Nicolas Mouquet from France, and then I'm here, hardly recognizable. I've changed in this other working group, not that much. John Chase changed a lot more. I think he would think the other way, but anyway. So this was the early days. This is kind of more recently what we've been thinking about, and what we've been thinking about that's really intriguing is to connect meta-community theory to the emerging statistics. Let's call them the next-gen statistics, using JSDMs. There's more people involved, but two of the important leaders in this area are Nerea Abrego and Otso Ovaiskanen. What this group of people did was to try to link these statistics to these sets of ideas by developing visualization and kind of hypothesis-driven methods of looking at these statistical models. And I'm going to skip over that, except just to say that we can look at how each species individually contributes to each of the supposed filters that we talked about. The spatial filter, which is influenced by dispersal, the environmental filter, which is determined by the suitability of the local environment, and the interaction filter, which is presumably related to species interactions. And we can do that for each species, but what we were also able to do that was find a way to calculate it for each site. So what this graph tells us is that there are some patches, let's call them lakes. In here, they're just theoretical, but to make it more tangible. There are some lakes that are occupied by plankton, which are the best ones for the local conditions in that lake. There are some lakes who are not occupied by those species. In fact, they have species who more or less are not very good under those environmental conditions, but they're close to other lakes where these other species are doing better, and they exist perhaps temporarily in these places. So you might guess that these lakes would be very isolated or something like that. And then there are some lakes that are where the species combinations have nothing to do with the environment or nearby lakes. They just have to do with particular species combinations that seem to be more likely or less likely to occur. We might think of this as being like keystone species. It doesn't matter what the environment is or where it is. These species determine the composition of the community by their strength of their interactions. So that sounds great, and the problem is that these JSDM's models, the parameters they give, don't match the mechanistic theory. And so JSDMs are not easily connected to Elton and MacArthur, even though it seems like they should be. Why not? Well, these are statistical models. They're only as good as the data you feed them. And if you don't feed them everything, they won't be able to calculate exactly what you're looking for. And that's possible. The other important problem is the problem of indirect effects. And so they're easy to interpret if we think that all the species interactions are just direct. This species acts as a filter, but that's not necessarily what's happening because there are indirect effects. This is a fascinating indirect effect documented by Tiffany Knight, who's a Helmholtz fellow at Halle. And she found, and her colleagues found, that fish could influence the population, the birth rate of nearby plants, terrestrial plants. I don't know why these people even thought that this was something that could be studied. My imagination wasn't good enough to guess that this would work. But what they found was that fish eat larval dragonflies. And that means that the larvae that emerge, the dragonfly adults that emerge from these ponds, were influenced by the fish. And because they consume the pollinators, they affect the seed production of these plants. I think it'd be fun to see if you can have enough imagination to figure out how these plants influence the fish. But okay, I haven't done that. And so it's possible to do that when you have just 3 or 4 or 5 species. But when we look at realistic communities, we would need to tease out all of this for hundreds, thousands of species. And that seems formidable. So these indirect effects can give non-intuitive effects. Here's a study by the first person who tried to solve this problem, Larry Lawler, and I couldn't find a picture of him because he didn't get tenure despite doing this work. And last I heard, he became a furniture maker who was quite good, but not on the internet that I can find. He did theory and used data from MacArthur's classic studies to show that if we have a set of species that all compete with each other, and we can calculate these net effects that account for these indirect effects, at least one pair of these species become mutualists. They're shown here. And the reason they become mutualists is that they both compete with this species here. What this shows is kind of a simple idea related to these net effects, which can be summarized as the enemy of my enemy is my friend. So both of these have a common species that they compete with more strongly than each other. And so jointly they can suppress the species and therefore are mutualist for each other. That's great. If we can do this in general, we have the problem solved. The problem is that real food webs look like, or real interaction webs look like this. This is an interaction web that was sort of described by dozens of years of work in the Chilean intertidal in this paper by Sonia Kefi and her colleagues. And you can see that this is truly entangled bank of interactions. Trying to quantify and describe all of these connections between these various species would be Herculean or Sisyphean, maybe, in scope. So that's not really a solution. So how can we transition from these basic ideas that we've come to understand and deal with biodiversity on its own terms? So the work by Mathew Barbier and Guy Boonen provides a link to that, and I'm going to kind of not do them justice. But what they show is that if we make an assumption of disordered systems theory, basically statistical mechanics, these guys are both trained as physicists, we can make predictions by calculating a much smaller subset of mean interactions and the variance of interaction strengths, instead of having to specify all of them, and make reasonably accurate predictions about biodiversity. They also show, however, that we can kind of cheat a little bit on the assumption of disordered systems. No rational, empirical community ecologist would be happy with assuming that species interact with each other in random patterns. Elton already told us that they didn't, because they interact in food webs. And they show that you can kind of fudge on this. And so, for example, here's a graph that has we can think of it as two trophic levels. We can assume that the interactions up here are disordered, the interactions down here are disordered, and the cross trophic level interactions are also disordered, and get a modified prediction, which can be a much more accurate fit than possible. It's also possible to look at spatial patterns, meaning meta-communities. And here's a figure from a paper that I did with Dominique Gravel and François Massel, where we tried to do that. So the question now is, can we link this general approach that accounts for these indirect effects to these joint species distribution models? If so, we could apply this disordered systems theory and test it, and maybe find out that it can be useful. The first approach, shown on the left, is not very promising. In this graph, we take each particular interaction that in our model that generated the data, and compare it to what we would have estimated that parameter to be using the JSDM. Yeah, there's a correlation, but you know, this is not especially impressive. You know, if somebody said which species is the most important species in this set of interactions, yeah, you know, I don't think you would be very comfortable with that answer. What we did was, and this is work that I'm doing collaboratively with Mathew and Guy, is we asked, well, what if we take the mean here? So let's say the mean actual interaction strength and calculate the mean predicted interaction strength. That seems like a much more modest expectation, but we find that we can be quite successful at doing that. So each of these dots is a different community, and we find that regardless of how we do it, we get a pretty good correspondence between those estimates. We can then ask, what about the variance? So is the spread going this way related to the spread going this way? We find promising results. I wish we didn't have that little data point there. It's quite annoying. I think maybe we can do a lot more and see if it's an unusual event. But I feel encouraged by this. So this way of linking the statistics to the process through this disordered systems approach seems to me like a really wonderful possibility that could link theory and data, mechanism and pattern across scales, etc. And it's only possible, this work is even remotely accessible only in the last few years. If so, if we can succeed at this, then we can start to ask questions about environmental concern, like ecological attributes and things that affect human welfare, like understanding the consequences of, I'll call it biotic mixing, but what I'm thinking about is invasive species. This is where I'm currently working in. What about novel ecosystems? It's not something you think about until you think that humans are part of the ecosystem, but each urban city has a biodiversity spot with its own unique features. Responses to environmental change like habitat loss or climate change, and to incorporate this into the ways that human ecology, meaning policy, might work. So for me, I'm getting old enough that I can consider and maybe will be encouraged to retire at some point, and I will not want to do it. And that's because I think ecology is at this beautiful point of inflection with our ideas and our possibilities. And so we used to spend our summers in the Alps in France. When I was a kid, it wasn't quite this beautiful, but this picture just makes me cry. And the cool thing about it is that we can think about realistic levels of biodiversity. Right now, I'm working with a data set on the biodiversity of water treatment plants throughout the world. We have in the 100 water treatment plants that we have data for, we have 90,000 species in our species pool. That's crazy. And we can move from understanding the details to understanding these statistical properties, and maybe use that to enhance human welfare. And I thank you very much for listening to me, and I'm happy to answer any questions or listen to comments, whatever you do. I leave it to Monica.
Thank you very much. Thank you for your talk. And you said you will accept historical, biological, and philosophical questions. So
well, I'll take anything. I may not answer it, but just.
So you can raise your hands on the left-hand bar, or just put something into the chat, and we'll call you up. I don't see anything yet. You said that as long as you don't have all the data, you cannot be accurate. But when would be the time? When would you say you have the complete picture? Can you say that as a scientist?
Well, that's kind of the point. I don't think we will be able to say that unless we have new, for example, extremely high throughput experimental methods that are not apparent to me. Like with microbial ecology, you might imagine that you might be able to do that, maybe. But what this disordered systems approach does is it means that you don't have to have all the details. You have to have enough to get estimates of the mean and variance and some of these other statistical properties, and make predictions. It's an open question whether that will be sufficient or not.
Okay, thank you very much. I have one question in the chat. Nitra Naik, would you like to read it out loud, or should I read it?
It looks like we have Mathew Biddick.
And we also have Mathew Biddick, and I will give Netra a broadcast permission for the next question. Okay, so please, cool. Matthew, go ahead.
Can you hear me okay?
Yes.
Brilliant. Thank you for the amazing keynote, Professor Leibold. I'm also a biogeographer, by the way. I work on plants and ecology and evolution. So I have a few questions. I'll try to restrict them to just one or two so I don't take up all the time. But my first one was when you talked about Richard Law's idea of species composition and how it was kind of cyclically changing, and members that made up the composition of species were slowly replaced and then came back. And yeah, it kind of reminded me of MacArthur's turnover. And I wondered how does it really differ, I guess. I'm not really up to scratch on this theory, but I'm definitely up to scratch on the theory of island biogeography. So I just wondered how those two concepts differ, or were they just kind of independently?
They differ in that in MacArthur, it's not a deterministic process. you know, there's colonization, there's a random component to colonization and a random component to extinction. And if you don't, you know, and the species are not interacting necessarily in the simplest version of Arthur's theory. There's no interaction. There's no, you know, it's just independent events that lead to this turnover. And that's been understood for a while. The phenomenon that Richard identified, and he's not the first one either, is a completely different mechanism having to do with the way species actually interact with each other. You know, so it's, you know, the simplest scenario would be if species A always outcompetes species B, species B always outcompetes species C, but species C always outcompetes species A. And so if you were just to watch that, you know, sit in one place and sample, you would say A, B, C, D, A, B, C, D, A, B, C, D.
Right.
It'd be the other way around.
The turnover is deterministic and mechanism rather than stochastic. Yeah.
Right, and one might, you know, question whether that happens in nature or not. And I've been fascinated by that, and I think there's quite good evidence that it does happen.
It certainly occurs on islands, that's for sure.
Well, no. Do you have evidence? Do you know of evidence on islands that it occurs for this deterministic reason, or?
Oh, not for the deterministic reason. I just mean that turnover occurs.
No, the turnover. Yes, of course. No, that's a great question. Thank you.
Good. Maybe I will let Netra go, and then.
Yes, I will. Netra, you could turn on your camera and micro.
I have turned it on. Yeah.
Yes. Now we see you. Hello.
Hello. Thank you for the wonderful and very insightful presentation, Dr. Leibold. I'm an architect, and I'm working on climate adaptation for cities. And one of the parameters we are looking at is how to integrate biodiversity into urban landscapes or into urban streetscapes, neighborhoods. And as an architect, it's very difficult to understand what parameters need to be considered to integrate species into these urban spaces. So I'm looking at simulation models and reading research papers. So my question is, are there any models or simulation models which can help integrate biodiversity into urban planning or study what parameters need to be considered?
Yeah, that's a great question, and this is one of those things where I'm not going to be able to help you with like specific for urban landscapes because I don't follow that literature enough to really be on top of it. But it's one of those places where, you know, my argument would be that this is just the kind of question where what is happening in community ecology can help provide a solution. I don't know of anywhere that gives you the solution, and someone will need to do the work. But maybe someone has, and I just don't know about it. But I think I'd like to think I would know, but you know, it's not possible to know what everyone is working on these days. But yeah, you should, if you cannot find it, you should try to find scientists who, you know, and say, what about working on this together?
Absolutely.
I mean,
yes.
That's where science is these days. I don't think, you know, I think these working groups are some of them have been magical. I mean, the meta community group, the original meta community group that I described was truly, you know, the most wonderful experience of my career to work with these scientists.
Absolutely, yeah, I agree. Because while thinking about planning, we as planners, we are clueless at what data sets to look at to study biodiversity and plan for it. So yeah, I think there needs to be a lot of research in that direction.
Yeah, I'm gonna give you one name, and it's mostly a joke actually, but it's actually not completely a joke. I do know a really great community ecologists that I collaborate with. His name, ironically, is Mark Urban. And he has worked on urban ecosystems.
All right, I will read up on his profile. Yeah.
Yeah, and you should just tell him I sent you to him, and he can thank me later on.
Absolutely. Thank you so much for the reference. Maybe I will write an email to him as well.
Yeah.
Mm-hmm.
He's a very nice guy.
Wonderful connections. Matthew Biddick also wrote somebody else into the chat. Professor Monika Egerer would be his advice. And we have one more time, I think, for one last question. Hamza, I gave you broadcast permission. You please go ahead. Not reacting. His question was, what relationship can we establish between genotype, environment, and phenotype?
Well, there is one, and it. Yeah, well, no, I'm not being facetious. I'm being, I'm trying to. So, generally speaking, there is one. I think my feeling is that it varies a lot when we don't know exactly why it should vary. So in some cases, for some species, the relationships are weak. For other species, the relationships are strong. You can think about why that would be. Well, one thing would be plasticity, meaning non-genetic components of phenotype. But then that kind of just pushes the question, because then why should there be differences in plasticity between species? And presumably, if there are differences in plasticity, that's due to genes. And so it's due to a more subtle aspect of the genotype. So it's not very straightforward. It's, of course, fascinating. And this is why studying biodiversity is so cool, because you never run out of questions. Your job is always assured. You will not find yourself without a job.
But I'm sorry, we are running out of time, not out of questions. I would invite you all to keep in contact with Professor Leibold. You'll find his contacts in the participants list, I guess. So please just go ahead and follow up with him. And there's a lot to do, we've heard. We now have a short break. And then at 3 o'clock, we will continue with either the science talk or the open forum. You'll find it in the personal program. Please just make a choice and enjoy yourself. Matthew, thank you so much for this keynote lecture, and
thanks
all
for
the best.
Yep. So much fun.
Yes, please. And stay with us for a short time at least, and maybe you will get some more questions in the chat.
Okay.
Okay. See you later, all of you, and enjoy the rest of the program.